Hello friends, welcome to my YouTube channel. Myself Janvi. Today I have come up with part 3 of the topic on cell growth and cell division. If you have not seen earlier parts, please see the description for the link. Before I start today's video, if you like my channel and videos, please subscribe to it and share it with others. To summarize this topic or the course on cell growth and cell division, it is made into 3 parts. So watch all the 3 videos to cover the topics. Okay, in this slide we will going to recap about what we have studied in part 1 and part 2. Mainly in part 1 we have studied about a cell cycle and what are the stages which are involved in cell cycle along with the interphase, and we have discussed in detail about the mitotic division including prophase, metaphase, anaphase and telophase. In part 2 we have studied about a cytokinesis, means the division of the cytoplasm, and what are the regulation of mitosis, like what are the factors which will bring about mitosis like MPF factors and role of cytoskeleton in mitosis. And we also studied about the importance of mitosis, why the cell has to undergo the mitotic division, mainly nothing but what are the uses of mitotic division. Now we will talk about the meiosis and reproductive cycle. The meiosis is nothing but the reproductive cycle. The meiosis at the end produces 4 haploid cells. Haploid cells are nothing or give rise to gametes. This gametes helps in sexual reproduction. There are different kinds of meiosis. Terminal meiosis occurs immediately before the formation of gametes. Sporic meiosis takes place at some intermediate time between fertilization and formation of gametes. Initial meiosis occurs in some algae, fungi. Meiotic division occurs immediately after the fertilization and only egg will be diploid in this condition. Here we will talk about the meiotic division. Meiotic division contains 2 phases, namely first meiotic division and the second meiotic division. First meiotic division contains 4 stages, namely prophase 1, metaphase 1, anaphase 1, telophase 1. Whereas second meiotic division also contains 4 stages, namely prophase 2, metaphase 2, anaphase 2, telophase 2. Meiotic stage is divided into 2, first meiotic division and second meiotic division. First meiotic division is further divided into 4, mainly prophase 1, metaphase 1, anaphase 1 and telophase 1, which I have already discussed in the early slides. Whereas prophase 1 is further divided into Proleptotene, Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. I will study what are the things and what are the changes will take place in each stage where the cell will go from the first meiotic division to the second meiotic division. In this slide, we will going to talk about the proleptotene and leptotene stages. Proleptotene stage will almost resembles the mitotic prophase. In this stage, the chromosomes are extremely thin. They are long and coiled-like structures. Whereas leptotene stage, here the chromosomes are long thread-like shape. They have the specific orientation in the nucleus. Centrioles duplicates and move towards the opposite poles. Again, on reaching the poles, it will again duplicate to form 2 more centrioles of a single diplosome. Okay, with the help of the diagram, we will explain the zygotene stage of the prophase 1 of meiosis. You can see here, these are the maternal chromosomes and these are the paternal chromosomes. The chromosomes which come from the father are called as paternal chromosome, and the chromosomes which comes from the mother are called as maternal chromosome. The one important factor which play a very important role in zygotene stage is pairing of the homologous chromosome. What is the meaning of pairing? The 2 homologous chromosomes come close to each other. They come very close to each other and they start pairing up with each other with the help of the synaptonomal complex. This is nothing but the protein which will bond the two homologous chromosomes to one another. This you can even call it as a crossing over, which will take place between the two homologous chromosomes. And the point where the pairing of chromosomes will take place is called synapsis. Now, with the help of the diagram, we will learn about the Pachytene stage of the meiosis 1 of the prophase 1. In this stage, the twisting of the homologous chromosomes takes place. The twisting means nothing but the exchange of the genetic material which will already have taken place in the Zygotene stage. Here, the cut between the homologous chromosome is done with the help of the enzyme known as an endonuclease, and the again the joining of the homologous chromosome is done with the help of the another enzyme called ligase. The point where crossing over takes place is called as chiasmata. In the diplotene stage, you can see that chromatids of each tetrad will be clearly visible and synaptomal complex will be disappearing. Here, unfolding of chromatids takes place and RNA synthesis will also take place in this phase. In the diakinesis stage, I will learn about how the nuclear membrane starts disappearing and nucleolus also starts disappearing. The chiasma moves from the centromere to the end of the chromosome. The movement of chiasma from the centromere to the end of the chromosome is known as terminalization. With this, the prophase 1 and then the diakinesis, the prophase 1, which is very important, will end with this diakinesis. Next, the cell enters into the metaphase. Here, in the metaphase 1 of the prophase 1 of meiosis 1, here with the help of the diagram, you can see that the homologous chromosomes have arranged themselves on the equatorial plate. The spindle fibers are attached to the centromere region of the chromosome. These 2 points are very important steps in the metaphase 1 of the meiosis 1. Once the chromosomes have arranged themselves on the equatorial plate, they start moving towards opposite poles in the anaphase 1. Here, using the diagram, I am going to explain you about the anaphase 1 of the prophase 1 of meiosis 1. I have already discussed in the metaphase the chromosome align themselves on the equatorial plate. Now the chromosomes will start moving towards the opposite poles and the cytoplasm will start to divide. That means the plasma membrane will already start to divide in the anaphase with the help of the actin and myosin filaments. So these two are the important changes which will take place in the anaphase 1 of the meiosis 1. Telophase. Here the two cells separates through the cytoplasmic division. The nuclear membrane and the nucleolus appears and two haploid cells are formed in the first stage of meiotic division. That is first stage of telophase of meiotic division. Till now, what you studied is just a meiosis 1 division with 2 haploid cells in them. These 2 haploid cells are next ready for the second round of meiosis, leading to the 4 haploid cells with the same repeating stages of meiosis 1, but with different names like prophase 2, metaphase 2, anaphase 2, and telophase 2. That means at the end of the telophase 2, you will get 4 haploid cells. That completes the total meiosis cycle with 4 haploid cells in them. Okay, now we will come to an end of the cell division and cell growth chapter. So I have provided the links to the video in the description. Please go through this if you have not seen this video. Let's recap on what we have studied in all the 3 videos, starting with the part 1. So in the part 1, we have studied about the cell growth, the cell cycle, the different stages of cell cycle, the mitotic cycle and different stages of mitotic cycle, and also we have learned about few factors which play a very important role in mitotic cycle. In part 2, we have studied about the role of cytokinesis. We have studied about the regulation of mitosis. What are the factors which will regulate the mitotic cycle, like MPF factor, or you can see the nuclear envelope, which play a very important role in the regulation of mitosis. In the part 3, we have studied about the stages of meiosis and also the importance of meiosis. At the end, we'll just see what we have learned in this slides. First is the cell cycle, definition of the cell cycle, different stages of the cell cycle, and 2 types of cell cycle, namely mitotic cell division and meiosis division. In mitotic cell division, we have learned about prophase, metaphase, anaphase, and telophase, and also we have learned about the importance of mitosis and what are the factors which will regulate the mitosis, and we'll talk about the meiosis, importance of meiosis, and division, 2 types of division, first meiotic division and second meiotic division, where in first meiotic division you can see prophase 1, metaphase 1, anaphase 1, and telophase 1, whereas in second meiotic cycle you can even see prophase 2, metaphase 2, anaphase 2, and telophase 2. This will be the end of the cell division. Thank you.